going on. Cells divide, grow, and continue to make contact with each other. The ability of cells to communicate makes life itself possible. But in a complex creature such as a mouse, the communication networks are so numerous that analysis is very difficult. We'll be returning to the analysis of signaling in animal cells later. But science's most successful stab at investigating a stimulus response system and its genetic control has been with bacteria.
This bacterium is about 2 to 3 micrometers long, and it's swimming at about 40 to 50 micrometers a second. That means it's swimming at something like 25, 30 body lengths per second. Now, to put that in perspective, that would mean a man about 2 meters high being able to move at about 200 kilometers an hour. A very efficient way of moving. Now, bacteria don't just swim randomly. They swim with purpose. And if you look at this simple experiment, I've evenly suspended some bacteria in soft agar, and in the center of this plate, I've put something that they like, a carbon source. And we're going to leave this for a couple of hours and actually see what happens. After a couple of hours, you can see that the bacteria have actually swum towards the carbon source. Bacteria use this response to reach their optimal environment in the wild. And this could be something like your gut wall, if it's one of the pathogenic bacteria. It could be a root hair, if it's one of the nitrogen-fixing bacteria. Or it could just be the right concentration of light, oxygen, the right carbon source. The modern study of chemotaxis was really initiated by an American scientist, Julius Adler, working in the early 1970s. And he looked at the response of Escherichia coli to a whole range of different compounds to show that there were families of proteins in the E. coli membrane that were there only to respond to environmental stimuli. They've got nothing to do with metabolism, nothing to do with transport. And he was able to identify 3 of these families. For example, here there's the TRG protein, which allows response to galactose or to ribose. So we now have a model for the sensory receptor system in bacteria, but what does this mean to the bacteria themselves? If we go back to the microscope, and if we now look at Salmonella swimming freely in a non-stimulated environment, you can see that they change direction every few seconds. But if I give them something that they actually like, for example, serine, then you can see that they hardly change direction at all. And if I then give them something that they definitely do not like, in this case indole, you can see that they change direction all the time. In a non-stimulated environment, the bacteria change direction every few seconds. But give them something they like, give them an attractant, and you can see that they're swimming for longer if they're going in a favorable direction. So they're biasing that 3-dimensional random swimming in a favorable direction, so that they end up, as a population, in their optimum environment.
Special research techniques show up the flagella which push the bacteria along. The bacterium looks rather like a tadpole, but its tail actually consists of a bundle of about 10 flagella forming a spiral. This spins rapidly and acts like a ship's propeller. To change direction, the rotation is reversed. The bundle of flagella flies apart and the cell tumbles before going full speed ahead in a new direction.
What we have is the only rotary motor in nature. What drives the rotation of this motor? If you look at this diagram of a small section of the membrane from E. coli, we'll get some idea of what we think drives that rotation. Up here we have a model of the motor, and you can see you've got 2 rings of protein in the inner membrane. One of these rings of protein is attached to the flagellar shaft and apparently free to rotate in the cytoplasmic membrane. The other ring of protein isn't attached to the flagellar shaft, but is somehow held rigidly. Then you have 2 rings to get the shaft through the outer membrane and then the flagellum. experiments have shown that it's protons that are required for rotation. The protons come from the electrochemical proton gradient. A percentage of those protons can go through the bacterial motor, causing this ring to rotate. That rotation is transferred through to the flagella shaft, the flagellum rotates, and the cell is pushed forward. It's possible to make antibodies to the bacterial flagellum, and it's particularly easy because it turns out the bacterial flagellum is only made of one protein. And if you make antibodies to the bacterial flagellum and coat a glass slide with those antibodies, you can tie the bacteria down by their flagella to that glass slide. And we have some in this flow cell on the microscope that are already tied down by their flagella. And if we look at them down the microscope, you can see the cell bodies. And you can see that the cell bodies are actually rotating. And they're rotating in mainly a clockwise direction, which is equivalent to the flagella going in a counterclockwise direction. But every now and again, they're rotating in the opposite direction. Now, if I give these bacteria something that they actually like serine, if I switch on a pump that'll change the environment that those bacteria are tethered in, and look again, what you see is they've stopped changing direction. They're all rotating in the same direction. If I now change the environment again and give them something that they actually don't like, which is indole, what happens now is the cell bodies are constantly changing direction. They're flicking from clockwise to anti-clockwise.
From genetic studies, we can identify about 40 genes involved in swimming and chemotaxis. Those in red are the flagella genes. Those in green are the chemotaxis genes. They lie in 2 clusters on the E. coli genome. This combination of genetic analysis and biochemistry gives us an advanced picture of chemotaxis in bacteria such as Escherichia coli. Attraction is mediated through one of 4 receptor proteins, TAP, TSR, TAR, and TRG. Attractants bind either directly to one of these proteins or to a separate binding protein associated with the receptor. If, for example, the cell encounters an increased concentration of galactose, this binds indirectly to the TRG protein, and a signal is sent to the flagella to keep rotating counterclockwise. So, what carries the signal? Mutant studies have implicated 2 genes, the so-called Key-Z and Key-Y genes. But despite extensive genetic analysis and molecular cloning techniques, we still don't know what their gene products do. We do know, however, that a methylation system allows the cells to adapt to constant concentrations of any one chemical.
What we've just described is a complete behavioural system in an organism that's only a few micrometers long, able to respond to and adapt to changes in its environment. And try and imagine that from the point of view of a bacterium. Here you have an organism that's swimming in something that's equivalent to liquid asphalt and too small to actually measure a change in concentration across its cell length. It has to be able to measure a change in receptor occupancy.
So this little cell, 1/1000th of a millimetre in length, effectively uses its receptors to measure the concentration at different times as it swims along and calculates whether or not it's going the right way. Larger cells could have a more sophisticated direction-finding ability. These are amoebae of a slime mould called Dictyostelium. They're about 10 microns long. That's about 10 times larger than bacteria, and they move much more slowly. These cells are swarming across a Petri dish towards a block of agar on the left, which contains a chemical attractant.
There is the interesting question now as to exactly how the chemical does this. And without going through any of the complications, it really amounts to this. You must think of an amoeba of having a nose at both ends. And if the nose at the front end gets more molecules hitting it of this attractant than it does at the posterior end, then it knows that it should go in that direction. So that what it is constantly doing is it seems to be taking readings at its anterior and posterior end and deciding where the chemical is more concentrated. And it's very much the same as we would do if we want to locate a smell. We would take a whiff here and a whiff there, and then decide that this one was stronger than that one, and that would give us a guide as to where the bad smell or good smell comes from. We unfortunately do not have noses at both ends, and so we can't be quite as clever at it as amoebae can.
Slime molds are interesting because the amoebae aggregate to form a complex multicellular structure which looks rather like a slug. The fact that this slug-like creature can move must mean that the individual cells within it communicate in a coordinated fashion. We assume that this must take place via receptors. Cellular communication in higher animals is more difficult to analyze because of the complex variety of cells present. But again, biologists are looking for specific signals and receptors. One clearly identifiable feature of young animals is that nerve cell growth is a late event. The growth of a limb, for example, happens before the growth of a nerve into that limb. But what signals the nerve to grow into the limb? Dr. Stanley Cohen of Vanderbilt University was one of those working on this problem in the 1960s. People knew that rapidly dividing tissue, such as tumors, would stimulate nerve growth. As a biochemist, Cohen wanted to identify the material responsible. He thought it was probably a protein.
We could isolate what we thought was a protein from the tumor cells, which made cells grow, nerve cells in culture, in order to be sure that nucleic acids were not involved, which were present in trace amounts. We tried to destroy the nucleic acids by putting in ribonuclease, deoxyribonuclease, and a very potent nucleolytic enzyme, phosphodiesterase from snake venom.
A snake's bite paralyzes its victim. Venom is a rich cocktail of components. It's a good source of hydrolytic enzymes, including the RNA-digesting enzyme phosphodiesterase.
And when we treated our protein factor with phosphodiesterase, a most surprising observation was made. And that was that the phosphodiesterase preparation itself contained an extraordinarily potent nerve growth activity, thousands of times more potent than the protein factor we had isolated from the tumor. And the end result of that story is that the protein in snake venom causing nerve growth was a contaminant in the phosphodiesterase. What conceivably could be the connection between nerve cells, mouse tumors, and snake venom? There was nothing very obvious. The thought occurred to me, where does snake venom come from? It comes from a modified salivary gland. Well, when we had originally found this nerve growth factor in tumors, of course we ground up all other mouse tissues, liver, brain, and so forth, and never found anything in the tumor, but we had never ground up salivary glands, because everybody knows salivary glands just make saliva.
So Cohen injected salivary gland extracts into newborn mice to see if nerve cell growth would be promoted. Indeed, it was. But something else happened.
What happened was, in addition to affecting the nervous system, a totally unexpected thing happened. And that was the injected mice opened up their eyes much earlier than normal, as you can see, and their teeth erupted as compared to the control animals. This is the control animal, and this is the experimental animal. And this was a totally unexpected finding. So what we were interested in, what was the substance causing eyelid opening and tooth eruption? What was it, and how did it go about its business of making eyelids open and teeth erupt?
Cohen decided that one way to find out was to look directly at the animal tissues.
This is a control 8-day-old animal. This is a cross-section of the eyelid area. Upper eyelids, lower eyelids, cornea, and this is the outside of the animal. The eyelids are closed because they are connected by epidermis. If you inject a litter mate with the extracts, the eyelids are open. They must have opened up because these epidermal cells must have grown and keratinized much more rapidly than in the control animal. In fact, the control animal would look exactly like the experimental animal 1 week later. So what we were doing was speeding up epidermal growth. So that was a straight biochemical problem, involved purifying the extract till we had one component that had the activity. We were able to do that, and the pure component turned out to be a small protein about 6,000 molecular weight. We were able to sequence this protein, determine its secondary structure, and that is shown on the next slide.
Working out the sequence of mouse epidermal growth factor was a minor triumph for Cohen's group and excited widespread attention. In England, Harry Gregory was pursuing what was, on the face of it, a completely different line of research on stomach ulcers.
Well, this was following our work on gastrin, the hormone-stimulating acid secretion. We were actually interested in inhibiting acid secretion, and we decided to look for a natural inhibitor. And the best-known descriptive literature on this was for urogastrin. This was first proposed in 1938 and derived from the observation that pregnant women don't get ulcers. The physiologists at the time soon found that extracts of urine would inhibit acid secretion, and this was fairly well defined. So we moved into this area and began by collecting vast amounts of urine, which is quite a difficult task. And we purified the material from urine purely as an inhibitor of gastric acid secretion. And eventually we got something which was highly purified, and we established a structure of 53 residues, single chain, 3 disulfide bonds, which was quite unique at the time. There was no other molecule like that at that time. And then at that time, Professor Cohen published his structure of mouse EGF. And by looking at the literature, it was immediately apparent that we had a very strong connection.
Of the 53 amino acids, 39 were common. The activities were identical.
We checked out the levels in human fluids, and EGF appears in all human fluids, some to in very high concentration. I mean, human milk has got concentrations of up to 100 nanograms per mil, so the human infant is taking on board a large amount. Prostatic fluid has a large amount, saliva, gastric juice. Every human secretion has EGF present in it.
So EGF is everywhere. But where does it act? This is mouse epidermis. If we stain for cells reacting to EGF, we can see that they're in the basal layer, where growth occurs. This picture is even more striking at a hair root. Again, the EGF-sensitive cells are the growing cells at the base of the hair. Cohen's next step was to follow the fate of EGF biochemically. Cells like these kidney cells derived from a marsupial rat can be grown in quantity. Cohen, in fact, used epithelial cells and incubated them with EGF tagged with radioactive iodine.
At this point in our studies,